So today we will be discussing with Dr. Patrick Tan his recent PLOS ONE Lab Protocol article entitled Use of the Waxworm Galleria mellonella larvae as an infection model to study Acinetobacter baumannii and the associated protocol in protocols.io. Dr. Patrick Tan is a molecular microbiologist committed to solving the global crisis of superbugs and antimicrobial resistance through innovative research strategies. His team has undertaken impactful research to explore novel antivirulence drugs, which has been published in many prestigious journals. He has secured various national and international research grants to explore new strategies to combat superbugs and antimicrobial resistance. He is regularly invited as a plenary and keynote speaker at international symposia on superbugs and antimicrobial resistance. Patrick's team established a research facility at Monash University, the first one in Malaysia, to rapidly screen the virulence of pathogens and to test the efficacy of new antimicrobials using moth larvae as animal models. His team has developed methodologies for rapidly detecting contaminated eggs and chicken in a user-friendly yet cheap format. This innovation won silver medal awards at the International Innovation Exhibition 2023 and the Malaysian Technology Expo 2023. He is a member of the Malaysian Society for Microbiology, the Malaysian Society for Molecular Biology and Biotechnology, and the German Society for Hygiene and Microbiology. Patrick, thank you so much for being with us.
Yeah, thank you very much for the kind introduction. Yeah.
So to kick off, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yes, thank you. Yeah, so essentially, this protocol, maybe I will start off by giving a brief idea why we're doing this. So essentially, this protocol is developed in order to allow us to use Galleria mellonella as a model, infection model. So Galleria mellonella is an animal insect that do not require any kind of ethics regulation because it is an insect. So this allows us to screen through really, really fast and also in a high throughput manner various drugs that we want to test, especially in our case, antimicrobials. So this allows us to cut down on the usage of these more larger animal models before we get to a final lead. So this can also help to save and reduce this animal cruelty and all this and allow us to screen much faster before we go to a much better lead or much better compound. So essentially, this protocol that we have developed, it is I believe, one of the very few reports that provide a step-by-step guide on how do you actually rear and also maintain the Galleria mellonella, right? And how do we do it in a very cost-effective manner without using any specialized equipment. And more importantly, in this particular paper, we also provided a protocol on how we can actually study the transcriptome of the bacteria that we are using. So before we developed this, we were looking around and we couldn't find any useful paper that can provide this. So yeah, essentially this protocol can be used for anyone who is interested to grow and use this larvae and also use it for infectious studies and also screening for antimicrobial.
So as I understand, as you mentioned, this is probably the one or just there are just a handful of protocols that describe the rearing of the waxworm larvae. But so to help understand better the significance of your optimization and development, can you briefly describe what was the state of the art when you decided to optimize the protocol?
Yeah.
How people reared the larvae and why those protocols needed optimization for your purposes.
Yes. Yes. Okay. Thank you very much. Yeah. So essentially, what we noticed from most of the papers that was published, usually when they want to run this kind of experiments, they will just go to the shops and those hobby shops, for example, those aquariums and those shops that sell this. They do sell this as part of the food feed, the food or the feed for the fishes. So when they get it, and do the experiments, there always be variation because they are not rearing it themselves. So this is one of the biggest problem for us because in order for us to get reproducible results, we need something that is a very highly controlled environment. And then we look further into papers that actually describes the procedure for rearing, and we found that they often need some kind of incubator, and all this costs quite a lot for us, especially in we wanted to keep this as easy as possible and as cheap as possible for people to do. So yeah, so that's basically the state of the art with regards to the rearing procedure. And also we noticed that there was really very few, in fact I'm not sure if I found any, on how you can actually recover the eggs, so that you can get the subsequent rounds going, right? So of course in certain countries there are already some companies selling this as a control, but in Malaysia and many parts of Asian countries I don't see this happening. So yeah, we decided to start this off our lab.
And for the user, when they want to implement your protocol, what are the most critical steps?
Right. So essentially, the most critical is the first is of course the housing for the larvae. How do you want to grow them? How you want to contain them? And we have to of course keep it in a temperature regulated manner. So that's I would say the most important consideration if you are considering about getting the larvae and setting it up. The other consideration, which is more for people who are interested in actually using our protocol to study the transcriptome of the bacteria, we have developed, I believe, one of the very first protocol on how you can actually separate out the bacteria from the host. Because for transcriptomic studies, oftentimes people do what we call as a dual RNA-seq. Basically, it means that you combine, everything that's there, the host and the bacteria, you sequence everything together. But then the problem is because the ratio of the host cell is way much more than the bacteria. So when you sequence them, you get a lot of this that is coming from the host, but very little from the bacteria, which is a big problem. So we solved this problem by coming up with a new centrifugation technique that can selectively not remove, but selectively enrich the bacteria. So, and this we showed also in the last part of the paper that when we check for the RNA, that really indeed we get a really nice, enrichment for the bacteria. So two parts. One is the rearing part, and the other one is for the transcription part.
And as you were optimizing and developing this method, was there anything that you tried that didn't work? I understand that you wanted to make this method very transferable, so cheap, so people anywhere in the world could easily, without any costly equipment, uh, establish the culture. Uh,
Yeah.
what are the things that you thought, you know, were intuitive, but at the end you decided not to include them because they didn't work?
Okay. All right. So essentially the optimization part, we have to start from really from the scratch, right from the very beginning of the diet. So we look into the existing methods. They use quite expensive foods. So we have, and some of them we couldn't find it in Malaysia. So we have to come up with our own defined diet. And one of the items we found that worked quite well, although I wouldn't say it's the cheapest, but it works pretty well. It's a good balance between giving good food and keeping the cost low. It is actually the cereals that is used for babies. So it's developed by Nestle. That is a commercial food, and we can use that, mix it with the honey. Um, it doesn't matter the grade of the honey, the cheapest honey, it won't work. So that's the first part that we have to optimize, trying out the different ones and see which of it actually support the growth and to a sufficient time. Because normally for infection, we need at least about a week or 2 weeks. So it should be able not to kill the not to kill the larvae within that time. Also, we have to optimize the housing and rearing conditions, as I mentioned earlier, suitable glass jars and all this to ensure that the eggs do not escape out. And also, we also noticed that Galleria is able to actually bite through plastics. So our first attempt on using plastic containers was a bit of a disaster because they just chew through and they just start running everywhere. So we have to use a glass jar to prevent that from happening. But with glass jars, of course, you have to make some holes for the air and all this, and so we also know we have to make some optimization from that end to make sure that eggs don't fall off because they are quite small, and they usually tend to locate at the top of the jar. The other thing that really I think people need to pay some attention to is the injection. So although this is not really related to the rearing, but it's related to the infection part if people are planning to use this model. So we have also highlighted in our paper, even with a picture to show how you can actually immobilize these using just a tip. So just use a cut fiber tip and you can immobilize this Galleria in there, and then you can inject it precisely at the location that you want to inject it, which is usually the legs. Yeah, I think the last one is of course the protocol on the how to establish the RNA extraction, but I think that's the typical RNA stuff, so I wouldn't go into that. Yeah.
And so you use the larvae to study as a model of infection of Acinetobacter baumannii. But
Yeah.
and I don't know whether you have the answer for these or it's just mere speculation, but how transferable or what other bacteria or how could these be implemented to study infection by other human pathogens?
Yes. In fact, this is very transferable. We of course we are not the only one have tested this on other bacteria. There are many other studies as well that has reported this on other bacteria. For us, we have actually tried this on other bacteria as well. We have injected Pseudomonas and a few other bacteria, and it's quite transferable. The technique remains the same. All you need to change is what you put into the syringe.
Okay.
So the rest of the things is pretty much the same, and we can follow. So it is quite a modular technique, I would say.
And I mean, developing a method from the scratch requires quite amount of work. But you went a step further and made the protocol available in the open access repository of protocols.io and then submitted it for peer review to PLOS ONE. What were the motivations to do that, to openly share it and then to have it peer reviewed?
Yes. So essentially, the idea is that we want to be to share our protocol in a very transparent way with everyone that is in the community, in the scientific community, that they are interested, to use this for their own research. And most of the papers, they are having just a short, tiny method section, right? Then they will just say briefly, we do this, this, this. But if you want to implement this in your lab, you can't be following that because you will have a bunch of much longer protocol. So this is where we come in, and we thought, hey, why not we just be transparent, let everybody have this opportunity to use, because the Galleria is up and coming model that more and more people are using. So and also why we want to make it peer review mainly is because two reasons. One is we want to ensure that our protocol is robust, because someone can independently audit it and say, okay, this is good enough, or maybe this part is not clear, which we actually got some of this feedback from the reviewers. We incorporate that into that our protocol, and I think that helps to make it much clearer for people who are using it for the first time. And more importantly, also this allows my PhD student who co-developed this to get due recognition, in a proper way. Yeah.
Yeah, and you mentioned two things that are important that I would like us to elaborate. So the first one is, I mean, you mentioned that the peer reviewers help you clarify some aspects of the protocol. Can you tell us more about that? Like how the feedback that you received from the reviewers shaped the final version of the protocol? That's point one. And point
Mhm.
two, having this peer-reviewed paper published, what has been the significance of that achievement for you and for your more junior colleagues?
Yes. Okay. So the first one is how it has shaped. Essentially, as I mentioned earlier on, it helps us to clarify certain sentences. So the how we express the sentence, maybe it is very jargon to us. We understand how it is, but when we put it out there, for someone who hasn't looked at it before, it becomes clear that okay, we need to improve on the sentence, on the construction. But it didn't change the protocol at all. It's just the way how we sequence it, how we express our sentences. So I think if you ask me in short, I would say it makes the whole manuscript clearer and easier to understand for the readers. To the second question on what's the effect, myself and also more junior colleague, I would say this protocol actually has allowed me to highlight our lab expertise to other researchers that's interested in this area, in using the Galleria and also in this area, and it also helped me to build new research network. So after I published this, I got another colleague of mine who wanted to use it for their screening of their bacteria, and we shared this, and I got another a couple of others, so it is useful and it's quite easy, when you want to communicate, say, hey, this is the paper, go check it out, the whole detail is there, and if you need further conversation, we can do that. So personally, I think it has been helpful in building up more the career profile, and I think people believe you more when you have a proper published, peer reviewed, and also in a top journal like PLOS ONE. Yeah.
And Patrick, thank you so much. I don't have any more questions, but if there's anything that we haven't touched that you would like to mention.
No, I think you covered pretty much everything quite seamlessly as well. Actually, it just flow through very nicely. So no, thank you very much for your time, and I really appreciate this opportunity to share and speak to you about what we have done in the lab.
Thank you so much.
Okay, great. Thank you.